Welcome everyone, I am Szalma from the Brain Imaging Center of the Research Center for Natural Sciences, Budapest, Hungary, and it is my distinct pleasure to present you our recent work on data-driven detection of dyslexia based on eye movement measures. First, I will discuss dyslexia briefly, emphasizing how the eye movements of dyslexic readers differ from control readers. Next, I will talk about the study design and the methods used to analyze the raw eye movement data. I will also present the achieved results to answer the two main questions of this study. How does letter spacing influence the classifiability of dyslexic and control readers, and what eye movement features are important for the classification process? Finally, I will draw the main conclusions discussing the impact and the limitations of the study. Developmental dyslexia is a reading disability characterized by decreased word recognition, speed, and accuracy, impaired fluency in reading and decoding, affecting between 5 and 10% of the population. Early detection of dyslexia would be paramount to mitigate some of the adverse effects. Natural reading relies on saccadic eye movements, and dyslexic readers have been shown to read slower by making longer and more fixations, more saccades, particularly more regressive saccades, with shorter saccade amplitudes compared to readers without reading difficulties. Dyslexic are sensitive to crowding, a perceptual phenomenon where the surrounding letters of the text negatively influence the perception of the central target. Although in theory this effect could be reduced by increasing the size of interletter spacing, results have been mixed regarding the relationship of eye movement measures in response to interletter spacing modulation. Although there are studies exploring the effect of interletter spacing on eye movement measures in skilled readers, to the best of our knowledge, none has been yet on dyslexics in a natural reading context. The 48 participants of this study included 24 dyslexic and 24 control young adults who were native speakers of Hungarian and had normal or corrected to normal vision. Dyslexic participants were recruited based on their official diagnosis of developmental dyslexia, and control subjects were selected in a way to match the dyslexics at group level. We recorded eye tracking data from the left eye using SMI eye tracker at 1250 Hz sampling rate. Prior to presentation and after reading of text lines, eye tracking precision and accuracy were validated. Participants read 250 sentences with 5 different letter spacings between 0.7 to 1.9 times the normal spacing. Each sentence was around 140 characters long, contained only words with lemma and word frequencies greater than 0.01 per million. After 25% of sentences, a test statement about the last read sentence was randomly shown, and participants had to report if the statement was true or false. Here you can see a stimulus example with a filtered eye movement overlay, red corresponding to saccades, green to fixations, and yellow to glissades. As it can be seen, reading is accomplished by a series of saccadic eye movements that are followed by fixations. Some of the saccades are followed by small corrective eye movements called glissades. To detect saccades, glissades, and fixations, and to extract eye movement characteristics, we used an adaptive algorithm. The set of 65 features included central tendency, median, and dispersion, the interquartile range, statistics of saccade, glissade, and fixation measures obtained after pooling of measures across text lines presented with the same spacing size. To classify dyslexic and control readers, a linear support vector machine was used. To find the optimal eye movement feature combinations, 5 different feature selection methods were considered. Forward sequential feature selection, backward sequential feature selection, recursive feature elimination, lasso, and elastic net. all of which were used to estimate best feature subsets and to generate feature ranking. Optimization of the C hyperparameter was carried out using a greedy grid search algorithm. To avoid overfitting, the whole analysis pipeline, including hyperparameter optimization and feature selection, was implemented using nested tenfold stratified cross-validation repeated 10 times. To evaluate the magnitude of overfitting that could occur as a result of hyperparameter optimization and feature selection, a standard stratified 10 times repeated tenfold cross-validation was also used. 4 metrics were used to assess the importance of eye movement features. Selection frequency describes how often a feature is chosen by a specific selection method. Individual classification accuracy of a particular feature was estimated by training a support vector machine classifier using that feature only. The added classification accuracy shows the average change in classification accuracy after adding a given feature to the combination of features. The leave-out accuracy decrease describes the average accuracy change when a given feature is excluded from the feature set. To evaluate classification accuracy and aggregate results of different feature selection methods, 3 metrics were used. Best subset accuracy considers the selected best feature subsets of each cross-validation iteration and averages these accuracies. Ranked max accuracy assesses the rankings of features generated by the feature selection techniques. The rankings of features were generated for each iteration by first considering the features with the highest ranking score and then adding the remaining features one by one. This metric is calculated by averaging the classification accuracy across the iterations and taking the maximum of these mean accuracies across the feature combinations with increasing feature number. Per iteration max accuracy also assesses the ranked feature combinations as the rank max accuracy. However, it takes the maxima across feature combinations and then averages across cross-validation iterations. Kappa analysis was used to establish whether the difference between 2 classification accuracies is significant. We evaluated which feature selection methods achieve the best classification accuracy across the letter spacing levels. With standard cross-validation, backward sequential feature selection achieved the highest classification accuracy for all performance metrics. In the case of nested cross-validation, we observed a lower disparity between the feature selection methods. The forward sequential feature selection method achieved the highest classification accuracy considering all performance metrics. However, the difference between the forward sequential feature selection and the worst feature selection method was only 3.5% for best subset accuracy and 4% for rank max accuracy. The effects of hyperparameter optimization were clearly beneficial in the case of standard cross-validation. After hyperparameter optimization, both best subset accuracy and rank max accuracy increased using backward sequential feature selection, but decreased using forward sequential feature selection. For the other selection methods, results are mixed. For further analysis, we only considered best subset accuracy performance metric, as it is the most accurate estimation of the classifier's generalization capability. The figure shows that both with nested and standard cross-validations, classification accuracy maxima for conditions spacing 2, spacing 3, and spacing 4 are achieved by forward sequential feature selection. For nested cross-validation, forward sequential feature selection reaches the maximum accuracy in condition spacing 5 as well. The highest classification accuracy obtained for condition spacing 4, 73.25%, did not differ significantly neither from the classification accuracy achieved by the default spacing condition, which is spacing 2 with 70%, nor from the lowest performance found for the minimal spacing size with 63%. To check which features are the most essential for detection of poor readers, we looked at features that were most often selected across the feature selection methods and letter spacing conditions in both cross-validations. To do this, frequency and individual feature importance metrics were simply averaged across letter spacing levels, while added accuracy and leave-out accuracy decrease were averaged, weighted by the selection frequency. Out of the first 11 features, 9 were the same in both cross-validation schemes. The most frequently selected features with nested cross-validation were median of saccade amplitude, median of forward saccade amplitude, median of fixation duration, and interquartile range of forward glissade amplitude. Note that interquartile range of forward glissade amplitude has a chance level classification accuracy individually, but combining it with other measures has a beneficial effect, while all other measures have a high individual accuracy. We also assessed how the most frequently selected features were distributed across letter spacing levels. The median of saccade amplitude shows a decreasing tendency over spacing levels 1 and 3, and stagnates after that, while the median of progressive saccade amplitude increases over spacing 1 and spacing 3, and then stagnates. The median of fixation duration was selected roughly the same number of times, between 35 and 50, for all spacing levels. In the case of the interquartile range of the progressive glissade duration, no clear tendencies could be observed. In sum, we have shown that even though maximum nested best subset accuracy of 73.25% could be reached in spacing 4, this performance is not significantly better compared to normal spacing and the worst performing minimal spacing condition. Out of the 5 feature selection methods used, forward sequential feature selection tended to achieve the highest classification accuracy. C optimization had mixed effects, but in overall it could not increase classification accuracy with nested cross-validation. The features found most important were generally similar for all spacing levels. The appearance of a glissade-related measure among the most important features indicates that there might be a difference between dyslexic and control readers at the level of the finest oculomotor processes. Our work in progress indicates that data-driven reduction of the number of features could reduce overfitting and increase test classification accuracy by 6%. Our preliminary results also suggest that linear classifiers might be sufficient for detection of dyslexia based on eye movement features. Classifiers with shallow nonlinearity do not improve the classification performance. However, these findings would have to be validated on a larger sample. A larger sample could be also used to test whether pooling the best performing features from different spacing levels could result in a better performing classifier compared to any given spacing condition, and the application of regression models could provide more detailed results on the relationship of reading abilities and eye movement correlates of natural reading. Finally, for early detection of developmental dyslexia, the translational capability of classification framework developed here for adults would have to be investigated on a larger sample size involving children. We thank Dávid Farkas and Dr. Dénes Tóth for their assistance in preparation of text stimuli and data collection. This research was supported by the Research Center for Natural Sciences, the Hungarian Scientific Research Fund, the Hungarian Brain Research Program, and the NeoPrism-C project. We also thank you for your kind attention. If there are any remarks, suggestions, or questions, we are happy to answer them online, but we can also be contacted via the provided email address.